From Microbe TV, this is TWiP, This Week in Parasitism, episode 107, recorded on the 7th of April, 2016. Hi, everybody. I'm Vincent Racaniello, and with me is Dickson Despommier.
Hello, Vincent.
And Daniel Griffin.
Hello, everybody.
How are you guys doing?
We're doing good, I think.
I'm
Daniel, how are you been?
I am doing well. I had quite an eventful last 24 hours, but I'm recovering.
And what was that event, if you might tell me?
I don't think Vincent knows about this. I tried to go to the opera last night. And,
I thought you were going to say I tried to go to Hamilton.
Actually, I have seen Hamilton, and it is fantastic, I must say. But no, so I tried to go see Madame Butterfly with my parents.
Yeah.
And right near the end of the first act, in the darkness, I hear someone shouting in a very loud voice, "Daniel, Daniel!" And I look over, and my mom has lost consciousness about 20 seats across the opera, and I go running over, and she's still unresponsive, is so for another minute or so, and she's now currently in the hospital being worked up.
And during this, the opera is going on.
Yes, actually.
Right. Were you in the orchestra?
Goodness.
You know, we were actually in row G, so we're really like right up in the front.
So they didn't stop for you, right?
So I ran, and actually the only way to get across was to run right across the very front of the front row, disturbing everybody, of course.
Wow.
And then I get there, and
nobody came to help or anything.
No, actually, it's great. You know, if you think about it, you've got a few thousand, and if you look around, mostly seniors, I'll say, in this large room. And so they actually have a Met physician who comes running down. And
so he came running down.
It was a woman. How sexist of you. But
well, I was going to say she. I was actually going to say she, but I didn't know.
Yeah. No, so they came running down. It was a woman. And my mother, became responsive, and we rushed her off to the hospital. And then as we're rushing her off, they called the physician because another person has lost consciousness over on the other side.
It's the opera. What opera was it?
As I was telling you, Madame Butterfly, which
there you go.
I think people were upset because it's really, it's not the most sensitive opera, right? It's about this American who buys a 15-year-old woman and marries her, and then goes off and gets himself an American wife, and she's with child in Japan and comes back with the American wife, and she commits suicide. She's so upset. I mean, yeah, it's a difficult topic. I understand people losing consciousness.
When the MD lady came down, did you say, "I'm a doctor"?
Yes.
So that she wouldn't tell you to go away.
Was your mother in any pain? I meant to ask you.
No. Fortunately, there was no pain, and
wow.
we hope everything's going to turn out well. So just a bit of a scare there.
You've talked to her today, I take it?
Yes.
She appears to be lucid and fine.
As lucid as ever.
Right. Well, that's not saying much, is it?
No. She's a rather lucid woman.
No, I would have gathered that.
So she was at her lucid.
Your mother, after all.
Her lucid, sharp baseline.
Right.
I wouldn't think I wouldn't expect anything less from your mother.
But you know what? The first thing I looked at on her white count was her eosinophil level, and it was 1%. I lost interest.
well.
We called the cardiologist and said, "You know what?
No
this is not exciting."
Yeah, right.
This episode is sponsored by the Auger Art Contest of ASM, the American Society for Microbiology. Submit a photo of your original auger-based art and a description by May 6th. Winners and select runners-up will be showcased in a gallery at ASM Microbe 2016 in Boston this June. And you will also get a free ASM book of your choosing. For full contest guidelines and terms and conditions, go to bit.ly/augerart2016. We thank ASM for their support of TWiP. Or lack thereof. No.
Now, let's be nice.
Now, we had last time a very, very interesting case from a colleague of yours. Right?
Yes.
In Switzerland, an ophthalmologist. Remind us, Daniel.
So my colleague, Flavia, had seen this woman in Switzerland. And it was a Swiss woman in her early 20s who had just returned after a surfing vacation in northern Africa. She described her accommodations as extremely rustic, lots of animal, insect exposure. The place was right on the beach. There was a lot of livestock. We described these sheep that they have, the Barbary sheep in the area there. Lots of flies, mosquitoes. And she did have some loose stools while there, and now she's back. Her left eye is bothering her. The eyelid is swollen. The eye is red. She noticed this when she returned, and she reports that she's seen things moving around in her eye. She saw the other ophthalmologist, the senior ophthalmologist, who told her it's all fine. Here's some eye drops.
You're just seeing things.
But now she goes back and sees my buddy, my colleague, Flavia, and he's doing the thorough exam. And I think we mentioned that he had actually seen several of these motile 1 millimeter long.
A millimeter.
So these are tiny, right? And they were, and I still remember him describing this because they appear to be heading towards the lacrimal duct.
Appeared to be, of course.
Yeah, who knows? I'm not sure how smart these are, but that was his vision, because now he is desperate to grab these before they vanish down into the tear duct. And I think we left people to let us know what they thought it might be.
And he plucked a couple of them off.
He actually plucked a couple of those off, and I got to see a picture of these.
Nice.
Of one.
Did you know, as soon as you saw the picture, did you know what they were?
No.
Right. That's what I thought.
I said, what's
what
that?
the heck is that? Yeah, well, I have my own suspicions.
We have some guesses.
We do.
Or I should say educated guesses. From Anne, who writes, "Dear TWiP team, regarding the Swiss student with swollen left eye, my thought is thalassiosis, perhaps caused by Thalassia calipedia. I did consider larval migrans and onchocerciasis. I found a comprehensive review paper on interdisciplinary perspectives on infectious diseases on ophthalmic parasitosis, a review article. How about that? They provide an extensive, exhaustive list of possible ophthalmic parasitoses, almost all of which involve internal structures of the eye.
Right.
Whereas Thalassia species infect superficial structures and feeds on tears. Seems poetic, stated that way. My only reservation is that Thalassia calipida does not, or calipida. How would you say it, Dickson?
I would say Thalassia first.
Thalassia. What am I saying? Thalassia.
Thalassia, yeah.
But it's spelled Thalassia,
I understand that.
Calipida.
That's close enough, I think. I've heard it pronounced multiple ways.
Does not seem to be endemic in northern Africa, although I found a reference for its occurrence in Europe, including Switzerland, but also consider the possibility, hey, maybe the whole trip is a red herring, who knows? Would also consider the possibility of a zoonotic infection, as several Thalassia species occur in animals in the region she visited, putting a plug in for fungal diseases for future case studies, by the way. And Dickson won't like that.
I'm not enthusiastic.
Are they parasites?
They can be.
Or viruses, right?
True.
Well, we have defined the case, the subject of this podcast as eukaryotic parasites. Of course, fungi.
Fungi are eukaryotes, but they're more plant-like than they are animal-like. They're closer to the bacteria than they are to the protozoans.
Yeah, you tell a fungus.
So I would, I've tried, but they don't listen.
Thanks, as always, for the engaging and illuminating podcast. Anne is a DVM PhD from Beaverton, Oregon. Or Oregon, sorry, I know, Oregon. I hear people already saying, Anne, it's Oregon.
Upset.
Oregon. Daniel, what do you...
Jeff writes, Dear TWiP doctors, a very interesting case indeed. When the discussion started, I started thinking maybe this was a case of Acanthamoeba castellani. But Daniel was going to say that she wore contact lenses. But as the description continued, there was the mention of flies, sheep, and the final mention of 1 millimeter long parasites being seen trying to get to the lacrimal duct, which sealed the diagnosis of Oestis ovis, which is quite common in several areas, including the Mediterranean. Definitely not your typical parasitic case, but quite thought-provoking. How is this treated? Keep this coming, Jeff. And Jeff Fairman is a PhD. He's vice president of research at Citrovax out in southern San Francisco.
Neat.
Estis ovis. Wow.
Never heard of it.
Dickson, could you take SJs?
I didn't. I've never heard of it before, to be very honest with you.
Then it must not exist.
No, I didn't mean that at all. So
it's in your parasitic book, I think.
It's in our parasite.
Yes, maybe it wasn't in yours, but it is in ours.
It is now. That's right. So SJ writes, "Hello, TWiPtastic."
TWiPtastic.
See, I have to get my glasses. I'll be right back.
Are you serious?
Okay, well,
Dead serious.
while Dickson gets his glasses, I will pick up the weight. I will read. SJ writes, "Hello, TWiPtastic trio. Sorry about the brief differential and probable typos. I'm typing this out in a hurry. That's okay because Vincent will jump in and fix the typos for us. Now, the differential, we have onchocerciasis, unlikely as immune response does not commonly occur prior to organism death. Loiasis, time frame does not match, but this is unlikely. Dirophilariasis, edema, symptoms of foreign bodies, presence of worm in conjunctiva, good one to look into. Ophthalmiasis externa, however, is my most likely guess. The presence of animals nearby, the rustic experience of sleeping outdoors, and the patient waking up covered with flies suggests the presence of fly larvae is likely. In addition, the often tiny number of larvae present can make it difficult to detect, allowing us to forgive the initial ophthalmologist having missed this one and treating it as standard conjunctivitis. Treatment involves removal of larvae added by anticholinesterase ointment to paralyze the larva. I look forward to the next episode. Thanks, as always." And now David is you want to read David there, Dick?
I can read David's.
Okay.
I can read David.
And 2 fingers to push it up.
Yeah, no. I was scroll down, as you would say. So this one is written by David, and David writes, "Dear..."
So 2 fingers and then you push. See how that, look at that.
"Dear professors, this week's case does not seem to be a very common parasite, and I don't have high hopes of getting it right this time. I did not manage to come up with a differential diagnosis, but will venture a guess based on my short research. Phthalesia. This parasite is transmitted by flies feeding from tears, and the larvae are deposited in or near the eye. The eyelid seems to be a reasonable target. The larvae then move to the lacrimal ducts to feed more. It seems consistent, but I could not find conclusive information about geographic range and size of the worm. Phthalesia calpida, that's right, the calpida, seems most common in humans and is found in Italy, which is at least Mediterranean-like Morocco. Phthalesia rhodesii is found in Africa and Europe. Suggested treatment appears to be a mechanical removal, which sounds as if there would often be just one worm, while in this case there were several tiny particles, so I am not very convinced of my findings. If I got this right, I wonder how it would be treated. Mechanical removal sounds horrible, but I am very squeamish when it comes to anything regarding the eye. Wiki mentions topical treatment with thiabendazole and cocaine on Wiki, and I found a veterinarian manual referring to ivermectin and the likes, and he gives a reference for that. Many thanks for the relentless transmission of stimulating podcasts. I look forward to hear the formal diagnosis of this case. Warm regards from Nicaragua, where April has started with very atypical showers after a dreadful heat spell."
Is that bad that we're relentless?
It's one way of putting it. I would say that we are conscientious in our
I think people like to hear repetition.
repertoire.
Not repetition. They like to hear, what do you call it? When you do things at a certain consistency. Thank you.
That's right. We
I'm
have
lost for words today.
a predictable pace.
Jeffrey writes, "Dear."
If anything, people want more.
Well,
Yes,
that's
they do
what we've
want more.
heard. That is what we have heard.
So we're giving them more every other week, right?
That's right.
Jeffrey writes, "Dear TWiP team, it is a cloudy -6°C here in Montreal. With no signs of letting up. Regardless of what any groundhogs may have seen, it seems that winter has stuck around for more than 6 weeks. As I finish up my U1 second undergraduate year of microbiology and immunology at McGill University, the TWiX series is only confirming that I picked the perfect major. My friends might find it bizarre, or as they would say in Montreal, bizarre. But podcasts on the weak critters of life and non-life make for good breaks after working through the CDs and ILs and THs of the human immune system. Wait till today's paper, where they get put into clinical context. Though it's not dedicated to parasitology, my introductory microbiology course, along with pages 1 and 2 of Google, This is someone who listens to our podcast. Has prodded me towards Thalassia calypida as my diagnosis for the case study of TWiP 106. T. calypida has been described in Mediterranean regions before, and its transmission through flies seems to fit with the accommodations of the Swiss surfer. I'm not entirely clear on what a blood test constitutes, which I should figure out for future TWiPs, but it seems reasonable that neither blood smears nor serology would give a positive result if the parasite remains localized in the patient's immunoprivileged eyes, as T. calypida does. Again, from Google MD, Florian seems to be on the right track with regards to treatment, as physical removal looks to be the accepted therapy. Loved the interviews with Dr. Libman from the MUHC, and loved Dr. Despommier's 2015 interview in Lucky Peach on vertical farming.
Aha.
Best wishes, and keep on twipping the light fantastic. What's Lucky Peach?
It was a group of people interested in urban agriculture, and I was fortunate enough to be interviewed by them.
Okay, that's it for our guesses. A rather sparse week.
I have a question for the presenter of this case.
Oh, that's me.
How would anyone from Switzerland decide to take up surfing? Where the heck would you go to practice?
Yeah, how did the Swiss? The Swiss do a lot of traveling.
That's true.
I was, I guess, 1988. I was living in Taipei, Taiwan, and I actually became friends with a lot of Swiss individuals who basically were living there. And I was surprised at how many Swiss and Australian and New Zealanders I would run into traveling throughout the world. So I think what the Swiss do is they leave Switzerland, they travel, they go and do these things.
But I mean, if you're going to take up surfing, and you live in Switzerland, that's going to be a tough nut to crack, I think.
Yeah, I think one of the advantages, right, as maybe our Swiss friends will know, is their economy is often doing quite well relative to other areas of the world. So
true. That is true.
When they travel, they they have the resources to go to a lot of places and do a lot of things. And
right. Okay, so back to the case.
So that's why she was
back to the case.
Back to the case. Vincent, have you got any guesses? Because I had some, but I'm not sure anymore.
Thalassia. That was my guess. Thalassia. Whatever you say it. That's what I think it is. Fits perfectly in my view.
Well, I would need to see that picture.
What picture?
The one that
Well, of course, that gives it away.
Not necessarily. You have to know what you're looking at.
Well, you would, because you're the man.
No, not necessarily.
You're the parasite man.
Yeah, but I'm not an expert in Thalassia. I'm not that for sure.
What are you an expert in? Trichinella?
Well, yeah. But I had other suspicions before the worm worked its way into the conversation, though, because, mites, for instance, bird mites.
Mites might do it.
Dermanisis, for instance.
Mites.
I don't know if you've ever heard of dermanisis mites.
I have never heard of
people who become allergic to mites, who have birds sitting on their windowsills and things of this sort, have been in contact with those organisms. They're not uncommon. And since she was surrounded by wildlife and domestic animals, the mite populations of those animals might be quite high. Okay. So I was thinking of something mechanical rather than transmissible. So the other thought was maybe because her eye was infected with something like a microbe, maybe this forced out her Demodex follicularums. Those are eye mites that actually live
they live
in the pockets of each hair follicle.
So are you saying we have a microbiome?
We have a microbe, a single species of microbiome, exactly right, called Demodex follicularum, and it's a very weird looking mite with a long body and a very short head.
How many do we harbor typically?
One per follicle of your eyelash.
I am going to look it up. How do you spell this guy?
Demodex.
Demodex.
Follicularum because they live in the follicles.
And I have, this I've never heard of.
Well,
genus of tiny parasitic mites that live in or near hair follicles of mammals.
They're not parasitic, though. They're actually performing a very useful function.
It says that they're parasitic.
I know
here.
it says. Where'd you look them up?
On the internet.
Yeah,
you know about that?
Which part of the internet? Which page?
It says Despommier.
I don't believe it.
65 species are known. 2 species living on humans have been identified, Follicularum and brevis.
All right.
both referred to as eyelash mites. And there's a picture here, Dickson, which you can look at. See that?
That is the one.
Can you see that one, Daniel?
And what is the size of that mite?
I will tell you.
Because she saw millimeter-long things. And I would guess that these things fall into that range.
0.3 to 0.4 millimeters.
That's right.
Does every human on Earth have one of these?
Apparently.
Where did you find this? I don't think I have them.
Well, I'm an expert at stuff like esoterica, and this is part of that esoterica.
They walk around at a speed of 8 to 16 centimeters per hour on your eye. And they come out at night.
See,
like vampires.
It's possible that some of them were forced out of their niche by the inflammation of her eye, which might have been caused by something else.
Older people are more likely to carry mites. About a third of children and young adults, half of adults, and 2/3 of elderly people carry them. There you go.
Right.
So not everybody.
Well, that's pretty.
I thought I didn't have this.
So just you and me, but Vincent is mite-free.
I'm mite-free.
That's right. That's right.
Mite-free.
He might be mite-free.
That's right. So that was my original.
That was your original. So I was trying to give, I'm trying to give people hints here, and I will say one of the people who wrote in, at least one, got it right. And I was trying to bring up a couple things. One is, I have to say that the person that ran across this article, the ophthalmic parasitosis, this review, it's a great article. And it's a good one, when someone has something in the eye to sort of, help go through the differential.
Right.
And there's a bunch of different categories of what could be there. So there's the amoeboid infections, right? The acanthamoeba would be the most common. And that's, at some point, maybe we'll do an acanthamoeba eye infection.
Right.
Then there's the worms. There's the nematodes. And those actually tend to have a length to them.
Like Loa?
Like Loa.
Loa. Exactly.
And
that was my guess last time when we did this, but you said it was too long. So
yeah.
Well, that's what I was trying to steer people, is that these were sort of very small and not necessarily much length to them. So try to steer people away from the different nematodes that might infect the eye.
Daniel, when you do a differential, do you do it all in your head, or do you sometimes sit and look at written things?
I always try to look stuff up.
Really?
I have I'll say the modesty that I can't remember everything. So I always try to put it up, and then I sort of go through, because in general, if it's not on your list, you're not going to think about it. Now, in this case, Florian grabbed what it was. He didn't necessarily have a list, and he handed it to the pathologist and said, "What is this? Yeah. What are these mobile things cruising around this lady's eye?" And I was sort of left in the same shock. Now, once you know what it is, then I try to focus some hints. So I try to tell you guys that they're not worm-like. They're more sort of small.
They're not worm-like.
There wasn't a lot of length. They're these small, like, particles.
Go on.
The other is, I wanted to bring up the proximity of flies and sheep, sort of make you think about. We call them ectoparasites affecting the eye. These are things that might be small, be mobile, but not worm-like and not amoeboid. And then you get into this differential of called the ocular infections caused by ectoparasites. And I think some of our people mentioned that because Thalassius is a worm, right?
Right.
And I would have tried to let you know that there was a certain length to it, or giving you the ground rules. They had. I have to say, in all honesty, when he presented this case, I was like, "Whoa, Florian!" I was a little bit aghast. What are these mobile things? So
you're going to show us the picture now.
So I'm going to show these gentlemen a picture, and I will give Dickson the job of describing what it was that was pulled out of this woman's eye. 3 of these were removed by Florian.
Wow. So this is a segmented, I will describe this thusly. It's an ovoid, segmented, that's very important, object. And this segmented object looks like not a mature thing, but rather a larval thing, okay? That's exactly what it looks like. So...
Doesn't it look like a maggot?
No, I'm not going to say that yet. I'm not going to say it yet, but I will say that I've spent some time in a Drosophila lab, and I've looked at all the stages of Drosophila, and this has some remote bearing similarities to a Drosophila larva as it feeds on banana media before it molts and gets involved in its life cycle to become an adult fly. There are no parasite nematodes that would exhibit this kind of symmetry because they are not segmented.
It doesn't
Right?
look like the thing I just looked up, Thalasia.
No, that's right. So it's not Thalasia. It's not any nematode whatsoever because it's segmented. So you can rule it out right away. And because of its,
Wow.
it almost looks like a, you know what a pill bug is?
Yeah.
It looks a little bit like what a pill bug should look like. And when it curls up, of course, it's round in its shape. But these little jobs, I've never ever seen any of them depicted before.
Well, he's right that one person got it.
So
About that, huh?
I think that's an amazing diagnosis, and I don't think I could have ever. But I'm now really confused as to why the senior ophthalmologist missed this, because those are not that unobvious.
Well, I think the issue is size, right? These are only about 1 millimeter.
Yeah.
These are tiny.
Still.
So when you hear the story of the woman, you've got to actually, I think, again, get on you and say, maybe there actually is something moving around this woman's eye. And what Florian, he actually taught me how to do this, is he has this technique where he takes the hand lens and uses an iPhone. And it allows you to really get a wonderful magnified view of the eye. And he's a very thorough guy, super nice. And he takes the woman seriously, and he spends the time, and then he sees them.
You just said a magic word. He took the woman seriously, because a lot of the people that you see in the clinical practice, and I was associated through a technician's job to listen to these stories by people who come in with these fanciful tales of parasitoses, which are all made up, and they're actually manifestations of their fear of being infected with something. So they have phobias. They have fear of being infected with insects. That's an entomophobia. There's a movie that was made recently called Arachnophobia, a fear of spiders that a lot of people have.
Like 20 years ago.
Well, it's replayed on television again and again. I keep seeing it on late night TV. Maybe that's true. But the point is that the phobia part of this plays a big role in a clinician's practice. So eventually, when they see enough of them, they start to assume that the real ones don't exist at all. And this is the one exception where it does actually exist.
So one person got it. That was Jeff.
Nicely done.
Estes Ovis. Very good,
Yeah,
Jeff.
that's great.
Nicely.
Congratulations.
Very good.
So yeah, as Dickson was starting to describe, when we when we remove the amoeba, the protozoans, when we remove the helminths, the worms, the flatworms and the roundworms, then we get to these ocular parasitoses caused by the ectoparasites. And as I mentioned, this article is great because they break it down geographically, and then they break it down into, Dickson brought up, you can get basically the mites, and you can often see little nits in the base of the eyelashes. You can even get these tiny ticks that actually infect the eye.
Wow.
They tend not to move around. They like to burrow in, and that's a bit of an issue. And in most cases, a perfect solution, if you can do it, is just to just manually remove these.
Right.
And that's what happened in this case. I mean, you can use the anticholinesterase ointment that was described, which, or you just basically. You grab them and use a very small tweezer.
How do you know if you get them all?
Exactly right.
So what he described was actually a hint, now that we know the diagnosis, that they were desperately heading towards, desperately heading towards the lacrimal duct. Because what they want to do, what they have evolved to have a tropism for, is in the sheep, they'll go up the nostrils and they'll actually develop in the sinuses, right? And then they will actually, they'll mature, and then the stuff will drop out. So they are, confused. There's a different environment in the human eye, but they're the same tropism to get into the lacrimal duct, to head towards sinuses. And some cases, people come in with not just the irritated eye, but a sinus fullness, and it's maybe a more advanced stage.
So they can reproduce in human sinus.
So
I don't know if they're able to go fully through, but they can continue farther in the maturation cycle.
I thought the lacrimal duct led to the lacrimal gland, which is a dead end.
No.
There's no way to get from the lacrimal duct to the sinuses, is there?
No, it actually goes down into the nose, yeah.
It goes.
And where does the fluid for the lacrimal gland come from?
Well, so your lacrimal gland is up high.
Okay.
And it's basically, there's a flow of tears that flows across the eye, comes into the lacrimal duct, drains into the nose.
The duct.
And sometimes that lacrimal duct will get actually occluded, and then the tears actually come down your face, just the normal tears.
All right.
So there are some rare cases of avian influenza virus infections in people working on chicken farms, where the infection starts in the eye, because there are alpha-2,3 sialic acids there, which the viruses like to bind with.
Oh my God.
It drains down the lacrimal duct into the nose and then into the tract, because in the upper tract, not so much 2,3. Okay,
Interesting.
so if you start in the eye, you can
That's interesting.
H5N1 and so forth. You got it?
Ha.
Got it.
Yeah, I do now.
And I show a picture of that to
It's
my
becoming
class,
clear.
the lacrimal duct. So
Right.
that's why. Nice.
What an unusual case, Dave.
And so,
Thank you for stumping
just
the
by
stars.
looking at the picture, that's it.
No, I have to say I was stumped too. I gotta look this up. What is this that you're
On car talk, guys, they used to say stump the chumps. Well, we're not chumps, but you certainly did stump two people that have a broad knowledge of pathogenic organisms and infectious processes, and I was absolutely
And one person who
befuddled.
doesn't.
Well, they had the advantage of time and looking it up, which we should have done also. I guess.
Florian shared this with us, and it was a group of tropical medicine infectious disease
Nobody knew, I'm
physicians.
sure that-
No, we were all like, "What is that?" I got to look that one up. That was,
Right.
but yeah, it was a great case.
Neat.
All right, we have a paper
We do.
selected by the eminent Dickson Despommier. Are you
I
eminent?
did. Imminent, perhaps.
Imminent?
Eminent, I don't think so.
From Science Magazine, tuft cells, taste-chemosensory cells, orchestrate, speaking of opera, orchestrate parasite type 2 immunity in the gut.
Right.
And when I read this, I
Right.
thought of Edward Tuft. Unrelated, but
Unrelated.
Tuft.
That's right.
And the first author is Michael Howitt, and the last author is Wendy Garrett. This comes from School of Public Health at Harvard, Tufts Medical Center.
Tufts Medical Center.
They're very
How
appropriate.
interesting.
Weill Cornell Medical College here in New York City, the Broad Institute, MIT, Dana-Farber Cancer Institute. Wow, it takes a lot of people to do a tuft.
I'll say it was a tough
Tell me,
going.
Dickson, you selected this.
I
What
did.
What attracted you? What
Oh.
chemo attracted you to this paper?
Well, first of all, I've been working around this issue most of my adult life.
What's
with
this
regards
issue?
To Trichinella, the mechanism of immune expulsion of parasites from the gut tract. So
Immune
this
expulsion.
article deals
I'm
exclusively.
glad you said that, as opposed to
with
physical expulsion, right?
Well, no, it has to be immune expulsion. So
He doesn't.
for
get
many,
jokes
many
today.
years,
He missed that. It's okay.
I know that's okay. But for many years, what you would call mainstream immunology back in the 20s and 30s refused to acknowledge the fact that the immune system could be directed against such large things as worms. And so people who worked in this area, this immunoparasitology area, either worked on protozoan infections, and they too had they were dealing with large organisms compared to the bacteria and the viruses that became known. And so there was a struggle to bring into sync the knowledge regarding immune expulsion mechanisms for bacterial, viral, fungal, and protozoan and helminthic infections. And this article seems to have found a bridge which allows for speculation regarding an overall process that it's like immune surveillance technology, which involves taste of all things. And, we sort of taste our food and say, I don't like the way this tastes. Can you imagine your gut tract encountering a microbe that doesn't belong there and saying, does this taste funny to you? Or
Taste chemosensory.
that sort of thing. And so here we are.
Funny to talk about taste in the gut.
We've got exactly right. And that's the other thing. Here's a cell that belongs in your mouth and lives in your mouth and does the right thing in your mouth too.
Mm-hmm.
And it allows you to sense all of the flavorings of your food.
So it's the same sensors that
It's
you
the
have in your
exact
mouth,
same
but they're in their gut. And of course, you don't taste anything because they're not wired the same way as the
Right.
ones in your mouth.
But I
Interesting.
wonder when you eat
It's like
whether these
recycling.
interleukins are involved
It's
in.
using one thing for multiple purposes.
Well, I like that a lot.
And you like that.
I
The
like that a
interleukins that are involved, they might be involved in taste too.
They're wild.
So this is so they have a nice way to introduce this. They introduce the fact that our gut is full of microbes of all sorts, viruses, bacteria, parasites, and then there's an epithelial layer that keeps them out. And we know innate immunity is important
We do.
for keeping this barrier, like toll-like receptors sense them, and when they get too close, they get repelled. And then they say, but there are also other things in there, and that's
right.
one of them are these
But look at how they found this. So even how they found it is remarkable.
I'm getting there.
Okay. I'm excited. No, I'm really
So
excited.
they say beyond pattern recognition receptors like TLRs, hosts monitor the microbiota by using these guanine nucleotide binding protein, G protein coupled receptors.
Yes.
They have an external domain and then an internal domain that's linked to a signaling pathway. All right. And this is many taste chemosensory cells have these receptors that are involved in sensing taste.
They do.
So they're like, and tuft cells are one of them.
Right.
Tuft. These receptors are on tuft cells, and they wonder, are these tasting the gut microbiota in some way?
Was there a hint that they might be? Because they had 2 strains of mice, right?
No, even before they had 2 strains of mice, Dickson. This is the introduction of the paper. So before you do any experiments, why would you even do them to begin with? Because it turns out that microbial products are sensed by GPCRs, and it turns out that tuft cells have GPCRs in them. That's why they said, let's look to see if these tuft cells are important, okay? I know you want to go right through the experiment without giving any background to the listener.
Well, I looked at this as a sort of, I did A and then D and then F and then Z.
Well, that's in fact the way science goes, but in a paper they rearrange.
When I put the paper in there, on
Yeah,
A,
of course.
B, C, D, E, F.
We were talking about with Firestein.
I know we were.
right?
Yeah, that's right.
How a paper is written in a way that's probably totally different from how.
Well,
it
this is
happened.
one of
No,
those
I was saying that over lunch yesterday. I'm doing this RNA-seq experiment, and I'm saying, well, you realize I'm doing that first, but that will come last when I write the paper.
That's right.
So Firestein said if they ever say in the introduction, we reasoned that means
Forget about
it just happened.
Forget about it.
So they actually say it is unknown whether. So I bet they first had the mice and they found this. You're right.
Exactly
Yeah.
right. So they had 2 sets of mice. One was from Jackson Labs, and one was bred in-house.
Correct.
And they looked for a cross between these 2 animals, and they found an increase in tuft cells of all things in the outbred strain.
The 2 strains differed to begin with.
They
right?
did. That's correct.
So the strain that they had 7% tuft cells in the gut.
The ones from Jackson Labs had a lot more.
How do you measure tuft cell numbers?
You just count them.
How do you know which ones are tuft
Well,
cells?
you can label them with something that lights them
they
up.
Probably have, maybe GPCR.
There you go.
They actually, so they do that in their early experiments. They label them, and they actually count.
Right.
So their mouse strain has 7%, and then the Jackson has 1%, which is lower, right? Now you may think 7 times lower is not a big deal, but
that's pretty low. So what was the difference then between the 2 strains, right?
So they looked and they found lots of single cell protozoa in their mice, but not the Jackson mice.
Exactly.
And they did scanning EM.
Right, which helped them a little bit.
If you had a scanning EM around, you could do that.
I guess that's.
See, I don't have one here. So what would I do? I'd say, Dickson, what do these look like?
Yeah, right. So I would say there were trichomonads of some sort.
You need to have a scanning EM to
you don't.
do that?
Of course not. You can see them moving around if you just crush the gut.
You can even do what they do is jump ahead and say they looked like trichomonads. Let's do some PCR.
They said in the low power microscopy, they could see some things that looked like trichomonads.
And then you could stain it and look for their axostyle. And would you do that there?
They actually did PCR to find out what they were. Trichomonas murus. Trichomonas murus. Why is the tri there?
Well, it has 3 flagella.
3 flagella. Wow. These are beautiful, by the way. They have a nice
gorgeous.
picture here.
They're gorgeous.
Gorgeous looking with that undulating membrane.
Yep.
Which goes along with the undulating fever. No, that's another.
No, that's another one.
So the trichomonads, they think, go along with having more tuft cells.
Isn't that interesting?
So then they decided to eradicate them. How would you do that?
Well, you'd use a drug. They put metronidazole in the water.
Was that a good idea?
It's a very good idea. Because trichomonads are facultative anaerobes, and metronidazole is a drug which attacks all anaerobic microbes.
So I would say it's a crappy experiment because it's eliminating a lot of stuff, even though the trichomonads are going down. Well, they said that too. So the trichomonads are going down and the tuft cells. So how could you get around that criticism?
But then
what would you do?
You put the trichomonads back in and see if you could
Koch.
Koch spatulates. That's
you grow them up and you put them in so you can grow these things, Dickson?
Oh, sure.
And when you give them back to the mice, I guess they fed them right?
They did.
What happened to the tuft cells?
Well, they only infected a portion of the gut tract, so they had a control. They had part of the gut tract not infected, part of the gut tract infected, and the only part that was had more tuft cells was the part that was infected.
Why do they only infect part of the gut tract?
Because they had an internal control from the same animal.
No, why? Not what was the use of it? Why would it only infect? Did they purposely?
They actually purposely.
Oh, they injected it into.
They injected
place?
it only into the last half of the gut tract. So
how would you do that?
well, you just open up the animal and take out the gut and pop them in with a syringe.
All right, so now we have this nice correlation between.
We do.
So what I would ask is what molecule in the trichomonas does it, but they didn't do that.
Not yet.
Because I'm a molecular biologist.
We're working on it.
Then they move into...
well, then they made a general statement, didn't they?
Helminths are the common eukaryotic... They go right to helminths all of a sudden.
Well, they said,
and of course, trichomonas is a protozoan,
that's
right?
correct, but it's a parasite.
It is.
So they said, what other gut-dwelling parasites can we use that are easily available?
And they're distinct from protozoa,
They could have picked Giardia muris just to make sure that another protozoan parasite did the same thing. They could have done that. They didn't.
I would have taken Racaniello.
Well, they did, actually, didn't they?
Was that your idea, Dickson? Did you tell us to use that?
I have to confess, no, it's not.
So they used a bunch of worms, including Trichinella spiralis.
They used 3 different ones.
Nippostrongylus brasiliensis.
That's right.
Which is a helminth.
Yes, and I dare you to pronounce the next one.
Heligmosmoides polygyrus.
Yeah, H. polygyrus is what everybody refers to it as.
What do these do?
Those are 2 hookworm lookalikes.
Hookworms, right. Nippostrongylus. Yeah. What's the other hookworm? Americanus, right? Or is that...
Look at you. You're
Nicator americanus.
You are
right?
almost a parasitologist.
Well, I have been doing this for 107
you know what?
episodes.
I wouldn't be an almost virologist, I'm afraid.
How many different types of genomes are there? Yeah, you got it. You can be a card-carrying virologist.
Or I feel like a trained seal every time you ask me that question. You throw me a fish,
and
I get it right. I always get it right.
Thing. So what do? Give these to the mice?
Yeah, one at a time, of course.
And how you would just feed them?
Yeah, they're easy to infect.
And what happened?
Well,
look at he doesn't even have the paper, and he knows.
No. This is down my alley. This is down my alley.
You like this stuff?
I love this stuff.
Yeah, I do.
Of course,
too.
I heard
pretty cool.
a career's worth of presentations on this without ever realizing which cell was.
Did you ever have a science paper?
Yeah.
Good. We should go over it sometime. Wouldn't that be funny?
It's okay if you'd like.
Good. We should do that.
No, it's an old paper.
So. Does that mean all old papers are no good?
No, it does not.
I was asked a question once at a Gordon conference by Michael Oldstone, a preeminent virologist. Mm-hmm. And I said, yeah, this is what they found, but those are old data. And he said, does that mean they're no good?
Yeah, right.
Yeah.
No, they were built upon by other people. No.
I don't know why I said they're old, right?
Yeah, my graduate student and I had our paper.
You're old and you're okay.
But you are right. There is a bias. You're like, oh, this was done back in the 70s. We have to reproduce it. No, I think there's
or not only that, they don't even refer to that.
Yeah,
that's the one thing that really gets me. You should acknowledge the old data as long as it's still valid.
So they give these 3 different helminths, and they all increase tuft cells. Notice I'm saying tuft because I like the word. They really
I know tuft. They're tuft.
Tuft cells.
Tuft. The other guy's name is Tufte, Tufte.
I know.
This is no E.
I'm very aware of this.
Tuft.
This
was done at Tufts.
That's why I say tuft.
Remember Tufts? They did the experiment.
Why do they call them tufts?
Because they have little on the top? They have little microprocesses at the end of the cell.
All right, next, Dickson, they say they're postulated to be chemosensory cells, these tuft cells.
Right.
But they're not known, right? So they wanted to know if perturbations could affect this expansion when you give them a parasite, right?
Exactly.
So, this is pretty funny here. Multiple taste sensory things sense sweet, bitter, and umami compounds, which I had to look up.
Right. It's a Japanese concept.
Do you know what it actually means?
Well, it means a pleasing set of flavors that combine together to give you a wonderful feeling in your mouth.
What was the word that they used?
That's what it is, though, really. Like, for instance, today
Not
for
sweet
lunch.
or sour. Yes. But it's
today I had
dumplings without pork.
I made a mistake, and I bought the ones that were not pork. They were just vegetarian dumplings. And they're totally different from the pork dumplings. There's no umami with the vegetarian dumplings.
But it was a wise food choice for you. I encourage you to continue
it's very healthy
avoiding umami and pursuing health.
These compounds, these sweet, bitter, and umami, they engage various receptors.
They do.
Which they found are present on tuft cells by staining, right?
They are.
And then they got a mouse that lacked one of these components, Gust-Deucin.
That's
Wow,
right.
what a name.
Gust-Deucin. That's
You know
right.
Gust-Deucin?
Well, it's
I
a
used to know him.
Oh, Gust-Deucin? Yeah, he was.
Fewer tough cells in Gust-Deucin null mice.
Do you know the two cells that make up your taste buds?
Only 2 cells? That's
They're
it?
called gustatory and sustentacular cells.
Oh my gosh.
Both of these, they're great names for cells, though I think they're wonderful names.
Gustatory. Well, good thing I ate lunch because I would be hungry
And
otherwise.
sustentacular, I like that name.
So fewer tough cells in Gust-Deucin null animals.
Right.
And then they used mice where they have GFP driven by the promoter of one of these tuft specific cells, so they can now look at them.
Right.
And then they have mice lacking this TRPM5, which is another one of these chemosensory receptors on the surface of.
Yeah.
They have mice lacking them, and in these mice, when you give them parasites, the tuft cells don't expand.
No.
Now, what's I going to say? TRPM5, wasn't that the sodium channel? That's sort of the end of the signaling pathway. So you have your receptor on the surface,
Right.
G protein, a phospholipase event, and then you activate your TRPM5. You get a sodium influx because the big distinction that we need to make is that we call these taste receptors, but taste is a psychological, right? It's an occurrence in the brain.
That's right.
And what these cells seem to lack, is basically a connection to the central nervous system, so that we can't have a conscious experience of the interaction in the gut, but we probably don't want a conscious interaction of what's going on in our gut.
How do we know that? People who are on parenteral nutrition, do they have preferences? Because they're fed through an NG tube that goes down into their stomach, and it avoids all these taste buds.
They definitely don't experience taste as we know it.
As we know it. But do they have a preference for different kinds of,
It's a good question.
parenteral
I
nutrition?
I think people are asking those questions. Does our gut microbiome stimulate our cravings, right?
Oh,
I mean,
of
people
course.
with
too.
a certain gut microbiome crave
Chocolate,
sugars or fats
yeah.
or
Or fats,
chocolate.
that's right.
So is there some level at which this is
Correct.
ultimately affecting our conscious choices?
This is translational medicine now.
Are the worms somehow via the tough cells and cytokines and getting us to...
This is actually a pretty good story, though, isn't
A
it?
symbiont is controlling our behavior.
Tell me about that.
Yeah.
Wow.
Tell me about
Could it
that.
be that Toxo also controls our behavior?
Not quite that way.
All right, next figure.
So the GFP, by the way, they use to show that the tuft cells with this TRPM, which you said is at the end of the pathway, right?
Yes.
Are in the distal small intestine, and it's in the epithelial surface, which is where you would want it to be. Now, these knockout mice, Dickson, you feed them TM. TM is the Trichomonas, Tritrichomonas murri.
That's right.
And then 36 days later, the mice lacking TPRM have much more worm burden than wild-type mice, which suggests what to you, Dickson?
Well, then, repeat the question.
It's okay.
I'll jump in.
Please
so
do
what,
jump
what
in.
we, and I think it's important that it's sort of at the end of the signaling pathways, because you don't have the TRPM5, you fail to have the output from these cells. And the output in this case seems to result in eosinophil migration and the many things that actually are effective immune barriers to the helminth. So what you've done is you've shown not only do the tough cells sense, but there's actually an output that's important for our immune protection.
Well, you were
against the
asking me
helminths.
about the output.
Yeah, so the tuft cells can sense the presence of parasites, and they multiply in their presence, and they somehow do something to inhibit.
They don't
parasitic
multiply
growth.
in their presence.
You get more tuft cells when you have parasites.
But they
right?
secrete an interleukin,
That does
which
what?
stimulates interleukin 25, stimulates interleukin 13, which then stimulates the tough cells to divide.
That's the same thing.
Yeah, but you have to get someone else to
getting
stimulate your reproduction.
that's fine. You're at the mechanistic level, but the end result is you have more tuft cells.
Yeah, the end result is you have more
That's
tough
what I said, right, Daniel?
No question about it. No, but how do you get more tough cells?
Oh, that's a great question. How do you, Dickson?
So that's
you
the
just
answer.
Said it.
That is the answer. So
these tuft cells are making interleukins.
Right.
And they show that in a series of very elegant experiments.
Exactly.
Right, Dickson? Let's see if we can go through some of those. So IL-25 which you just said, right?
Yes.
And what's the point of IL-25, Dickson?
Well, the
Tell me.
point of IL-25 is that that's produced by the tuft cells itself. It's a signal to other cells that control the growth of the tuft cells.
Because epithelial cells are a key source of the parasite-induced cytokines TSLP and IL-33 and 25.
Right.
They isolated the cells to determine the expression patterns of these guys. Consistent with recent reports, we found that tuft cells make less of these than other epithelial cells and are the main source of 25.
They are.
IL-25.
That's right.
So then they take wild-type and TRPM null mice. They put tritrichomonas into them. They measure parasite number and these 25.
Right.
And so wild-type... And so this is the interesting thing. Both wild-type and knockout mice are equally colonized, but in the end, the knockout mice have less IL-25
Indeed.
12 days after infection.
Indeed.
So you need TRPM5 to make IL-25. And IL-25 is a chemoattractant, you said?
No, it actually...
mitogen?
It stimulates an innate immune cell to produce IL-13.
Would that be a type 2 innate lymphoid cell?
It would. And that's what they said it is, at least.
Yeah.
And then that interleukin actually works on the tuft cell itself, but it also does another thing. It stimulates the production of goblet cells, which produce mucus.
Right.
So you have two things going on at the same time. You have tuft cell increases, and you have goblet cell increases. And at the third situation, you've also got specific antibody that's being made against this organism at the same time. So you've got a combination of events all coming together to produce the expulsion mechanism, which is complicated. It's not just like make the antibody, it'll stick onto the organism and kill it.
But it's more complicated than that.
It's even more complicated.
Antibodies are more complicated than that.
Which is why it took so long for this to come.
Don't use antibodies as a simple model.
I was using them as a viral neutralization
Yeah, but there are many ways that antibodies work. That's what I'm saying. You can't say one thing is simple and not the other. So IL-25 binds a receptor on type 2 innate lymphoid cells. Daniel, what are type 2 innate lymphoid cells?
Yeah, Daniel, type 2 innate lymphoid cells.
No, this is good because we're back to sort of the basic immunology that we talked about. And I try to give people a mnemonic for remembering this. And if you're, let's say, tuberculosis, I use that as the model. That's this type 1 reaction, which is a cellular response.
Right.
And then type 2 is more of a humoral. It's an antibody based. There's a whole set of cytokines, IL-10, IL-6, IL-25. What's the other IL that you just mentioned?
33.
33.
I actually, the other day when I was like, I need to keep this list straight in my mind. There's there's a group of cytokines. You only have so many cells. And what you're doing is you're shifting kind of the focus of the response to either be more towards an antibody base, which would be great, we think, for a helminth, right? A lot of eosinophils, a lot of antibodies to cover these big, large pathogens. But then when you have, let's say, a virus or an intracellular pathogen or something where you need cellular, you want to shift your energy that way. And what we've seen over time is sometimes the parasites like to shift things in a certain direction to thwart our attempts to clear them.
Yeah.
And sometimes, as we saw with Trichinella, right, they actually promote a type 2 because something about the eosinophils is necessary for them to go on to create this whole nerve cell business.
Very true.
So these type 2 lymphoid cells, they go up in number when you give mice Trichomonas murus. But if you use the TRPM null mice,
that's right.
the lymphoid cells do not increase.
Right.
So that is suggesting that the IL-25 produced by tuft cells is helping the type 2 innate lymphoid cells proliferate.
Indeed.
Right? And this receptor is the thing. And they add exogenous IL-25 in, and that fixes the problem.
Right.
Restoration of distal small intestinal eosinophilia and tuft cell abundance. So the eosinophilia is a consequence of IL-25. Is that what you said, or 33?
No, neither one. It's an IL-5 response.
5, and where does IL-5 come from?
Good question, Daniel. He's our IL.
No, actually, IL-5 is produced a lot by eosinophils.
Right.
So
but what recruits them?
I actually, when we went back to the TRPM5 release, it ends up, and I thought it was actually IL-33 and IL-25 that were promoting eosinophil expansion. But
But there's an IL-6, that's for IgEs, I guess I meant to that, because eosinophils and IgE are connected together in some way.
I think that's, I mean, IL-6 is another one of our Th2 cytokines.
Right. I misspoke. I
because they do in one of these experiments where they actually they give IL-25 and you see like a 3-4-fold expansion of eosinophils. And even in the TRPM5 knockouts that are failing to do this, when they give IL-25, you get it. So I think the IL-25 is directly involved in the eosinophil expansion.
So I wonder if
IL-13, as we see, is directly involved in, as you mentioned, as Dickson mentioned, I'm pointing to Dickson, in the tuft cell percent expansion.
So this is an aside, but not such an aside. It's apparent in a large population ecological / epidemiological study, no matter where you go, you will find a maldistribution or a skewed distribution of numbers of parasites per person for whatever parasite you pick. So schistosomes, hookworms, trichinella, even malaria. You will have some people that harbor loads of these parasites and other people in the same population that are genetically unrelated to those people. They don't harbor so many parasites. So at least in the gut-bearing parasite loads, maybe there's a relationship between people that have a lot of tuft cells and people that don't have so many. Maybe they should start looking for something like that and seeing whether you can correct it by simply giving the interleukin that tuft cells might produce if they were there. And that might be another treatment modality that you could apply to these groups of people that are compromised. No?
It could be.
No, I mean, I think that what Dickson brought up very early on was, being a molecular guy, he wants to know what is it on the parasites that triggers this whole cascade.
Yeah,
what are the tuft cells sensing?
Yeah.
Because I think people still, are not so excited about, we're going to give you a whole bunch of nematodes, a whole bunch of parasites, because we think it's going to help your health.
Well, you're thinking of giving
Crohn's disease
IL-25, right? You want to do
yeah, you don't know. But
do you do interleukin therapy at all, Dan?
There is some interleukin therapy that we do. But no interleukin 25 as of yet that I'm aware of. But no, that's not only do we give interleukins, and there was some interesting interleukin, even interferon treatments, but we often try to block these to block inflammation. Right?
And I thought the point of IL-25 was it stimulates IL-13, which is the growth factor interleukin for the tuft cell.
Well, that's the question they want to answer. Actually, they want to know whether IL-25 works on the tuft cell to make it expand, or via the innate lymphoid cells.
So who made the IL-25? I was under the impression that the tuft cell made the
tuft cells do. But the question is whether that then goes back on the tuft cell and makes it divide, or is it via stuff produced by the innate lymphoid cells.
So could you give IL-13 as a short circuit for a non-immune animal?
I don't know where
and get an expansion.
Where does 13 fit in?
So they do in figure.
Oh, that's later.
So in figure 4,
yeah.
they actually go ahead. They give IL-13, and even in the TRPM5 knockouts, you get tuft cell expansion.
That's right. And they did this actually in an organoid system.
So
they didn't actually do that in
is
mice.
a growth factor for tuft cells.
They make organoids, Dickson.
Right. Yes.
Which reconstitute all the epithelial subsets.
Exactly.
They make them from intestinal epithelial cell stem cells.
Right.
They differentiate them into a multi-level thing, right? A little balls, they're organoids. Right. We're actually doing this with brain organoids in the lab. That's why this is pretty cool. And you can do it with placenta. You can do the different layers of the placenta and infect them and so forth.
Cool. Very cool.
And they find in these organoids they have tuft cells.
Okay.
Because they are GFP positive using that promoter that we talked about before.
Yep.
IL-25 does not increase tuft cell numbers.
No.
But IL-13 does.
Right. So IL-25 triggers the secretion of IL-13, which then comes back and makes the IL.
And you know who makes IL-13?
Yeah, the
innate lymphoid cells.
The innate lymphoid cells.
So the tuft cells make 25. They stimulate ILCs. ILCs make 13, and then you get more tuft cells.
Isn't that interesting?
It's quite interesting.
Yeah. Fascinating. And so it fills in a bunch of missing data for these presentations that, like I said, I mean, I've spent most of my professional life listening to presentations as to what the immune mechanism for a particular worm might be that lives in the gut tract. And they were all missing part of the story because they didn't suspect that this minor cell type was playing such a major role. They did suspect, however, early on that goblet cells play a major role. But I think what goblet cells do is distribute the secreted antibody along the surface of the gut tract and allow it to come in contact with most of the worms that are living either loosely attached, like hookworm would be, for instance, or the adults of Trichinella, which might be embedded deeply into the.
Yeah, I think what really comes out for me from this paper is the complexity of the system. And when you mention these organoids, we think of the intestine is there to absorb our nutrients, right? That's what it's there to do. But there's really 5 major cell types there, and only one of them is the absorptive enterocytes.
Sure,
we have these enteroendocrine cells there, which sense that we've had a diet. They modulate motility. We have the wonderful Paneth cells that I think
cells
Dickson
of Paneth.
And the cells of Paneth. They're a sensory type cell. Again, they're using TLRs, things like that. They've got granules. They're sort of a cross between sensory and immune cells.
Right.
We've got the goblet cells, our mucus producing cells.
Yep.
We even have those, the mucus producing cells in our airways. And then we've got these wonderful tuft cells. So they're really a complexity to our gut.
A lot of stuff going on.
One more experiment, though. Very cool here. Listen, so it turns out that STAT6, signaling protein, an intracellular signaling protein that responds to receptors binding ligands and it changes nuclear expression. STAT6 is required for expulsion of helminths.
Huh.
All right? So, remember, they showed before in the organoids that IL-13 induces tuft cell expansion. In a STAT6 null mice, the tuft cells don't expand. So you need STAT6 for this effect as well. And they also look at the role of T helper cells by looking at a variety of knockout mice, which is really cool. So RAG2 mice, null mice, lack T helper 2 cells, right? Type 2 helper cells. But they do have intestinal innate lymphoid cells of the type 2 variety. They have no tuft cell increase during infection with the trichomonas. So you can tease out even further the pathways and the cell types that are important here. So tuft cells detect trichomonas through this receptor, this taste-chemosensory receptor.
Right.
And they make IL-25, which attracts and helps proliferate the intestinal lymphoid cells, which make IL-13, which then comes back and turns on the synthesis of new tuft cells
and goblet cells at the same time. They do both.
Wow,
lots going on down there. Rumblings.
Well, that's because we have all the genetic power now. We have knockout mice that we can use to do this.
Yeah.
We know a lot of the genes that are involved, which have been identified over the years, and you can do this wonderful genetic stuff and make sense of things.
Right.
Which people should do this for Trichinella. Oh, they did it in this paper, didn't they?
Right.
Would you like to have done this paper, Dickson?
Absolutely. I would have been joyful to be participating in it, of course. The results are very clear.
Taste cells in the gut.
Very clear.
Interesting.
Yeah.
Taste receptors.
Who would have thought?
I just wonder if you can feel something, Once it's in your gut, we think it's the end of it. There's no more taste but maybe there is, as we said.
You can continue to enjoy your meal.
Yeah, long after
for
it's
hours.
Being chopped
up.
The point
is that what allows us to enjoy our meal to begin with? I mean, I've seen all these wonderful movies about chefs and food and, Babette's Feast and Au Chocolat.
Like water for chocolate.
Like water for chocolate. No, but there's another one called Chocolat. And then there's Ratatouille, of course, as a cartoon. And you see all these fantastic plays on... You see food critics getting very upset with the fact that they put too much cumin on their whatevers. The point is that some people have more acute taste buds than others. And so what are they experiencing that we're not experiencing?
I think there's too much emphasis on food in our society.
You do think so?
It's ridiculous.
You think?
Look at all the restaurants. There are now TV shows left and right with people competing, even little kids competing.
I know.
It's ridiculous. I hate them. Just study and go to work. Forget the eating. Just ridiculous. It's way overrated. You know what the thing is, Dickson? You look at these restaurants. They're contaminating the environment. They're spewing out fumes with the stoves. They're using resources.
Is that what they're doing?
It's just they generate tons of garbage.
Do you like wine, Vincent? I know you do, so you don't have to say you don't.
I do.
I know
I do
you
like
do.
wine.
You know how much effort has gone into producing wines of various sorts? Of course you do. It's not. It's what you just said about restaurants is
Well, we
nothing
should get rid
compared
of
to
that
what
too.
they
Then
do.
okay, get rid of it all. Sure.
No.
Get rid of it all.
Let's do it in a way that's not harmful to the environment.
I do think that we harm left and right.
Let's do that.
For example, this morning I was looking at dry cleaning. Each hanger has a covering of paper.
That's right.
How many billions do we throw away every day of that?
Do you
It's
return your
ridiculous.
hangers to the
I
cleaners?
do.
I do
That's
too.
why they're lying on my bedroom floor because I haven't done it in a long time. But
Yeah.
I think this is obscene, all the waste. And you get a package from Amazon, cardboard.
No,
I mean, I recycle it all, but still, you just think of lots of people don't bother.
You are
It's
right.
crazy.
You are right.
Do you have another case for us, Daniel?
I do have another case.
Oh.
I was just going to suggest everybody just consume Soylent because it tastes horrible. It has everything you need.
Right.
But Dickson, the bottom line
Charlton
is
said that
what
first.
people like, we pursue it. That's it. We
Yeah.
pursue what we like, and
Yeah, I know.
it's a free economy here in this country. It's a capitalist society, so you have competitions. You have a lot of money put into chefs, and you are involved because you're doing vertical indoor farming and you're providing leafy greens for the restaurants.
Yeah.
I think it's just scary.
Scary. Okay.
You know that they're talking now about having a glut of oil because we're going to have so many electric cars soon.
Did you know that all of
no.
those, if you go driving to the airport and you drive past all those refineries, all those tanks are full now. Every one of them.
Daniel, did you buy the new Tesla?
No,
Did you order one, I mean?
No.
I know someone who did. Steve Chen.
They've had
Steve
over
Chen, the person who worked for me.
350,000 orders. It's going to take
Wow.
them 10 years to fill
I
them.
know.
They only make 50 a year.
Oh, they're
Oh
going to
my
make
gosh.
more than that. I can tell you that. They're going to make more.
Well, that's
BMW
nice.
is
Now they
making
can
an
ramp up.
electric
Do you know
car.
if this thing hadn't worked, this order, the company would have gone out of business because they were losing money hand over foot.
They made the other model as a demonstration
Yeah,
project.
but this had to work.
Well, they knew. Well, they've already tested it out. All they had to do was dumb it down now.
Well, they had. It doesn't have a dashboard.
I didn't know that.
It just has that little readout. All right, enough
Oh, wow.
of cars. This is this.
Okay,
week
so
in
let's talk about a
Parasites.
case. And I
Sorry,
have to
I'm
admit, I was going through the cases and I
You're
was
running out?
surprised that I hadn't actually presented this case
Okay.
before.
Oh,
This,
good.
Let's do it.
This is a fun case. It's another case from India.
A fun case.
It is yes, it's a fun
I
case.
hope it has a happy
Okay.
ending.
So this is the story of a 45-year-old gentleman, and he is from Assam, India.
Oh, I know
I think
where
we
that
talked
is.
about this
I know
before.
where that is.
So you think of
From
India
Assam?
as being a diamond, right?
North.
And then in the northeast of the diamond is this little bit of India that just kind of sticks out and surrounds Bangladesh.
That's right.
Right?
It's next to Bhutan.
They kept the nice highland area, and then they gave the swamps to
I'm sorry
somebody
to say
else.
that. It's a big valley.
And so this is a 45-year-old gentleman from Assam, India. Are you ready for this? 16 years of fever, abdominal pain,
Oh, Lord.
darkening of the skin, and yellowing of the eyes. And he reports that the fever occurs every other day.
This is what we call recurrent fever.
Undulant.
So every other day.
What
This has
kind
been
of
going
fever?
on for
We low
16
grade
years.
fever.
At this point, he just, so he's describing it. He's just saying, I feel
It
hot.
feels hot.
Every other day, I feel really hot, not so well.
Okay,
Darkening of the skin.
His skin is darkening.
got it already.
Skin is darkening.
Well, I know what that translates to in Indian. I know the Hindi word for that.
Ah,
Oh, is there? There's a Hindi word for this.
There's actually two Hindi words.
okay. Continue, Danny. Don't listen to him. He's just
Okay.
distracting.
And we asked the man about prior medical problems, right? And he says nothing out of the ordinary, just normal things that people get, right? He's had no surgeries, doesn't take any medicines. He has not seen a physician his entire life.
They don't exist in Assam.
No, actually, this
Is he
is
married?
very far away. He's come.
Is he married?
He is married, and he has kids.
Right.
Does he have extramarital affairs?
Not that he reports. Specifically asked about.
Is he AIDS positive?
He is HIV negative. He does not have HIV.
What does he eat?
He says he eats the normal things that folks eat. He eats
Meat?
vegetables and not
Might
so
be a
much
vegan.
not
Might be a-
no not so much meat. Yeah,
Mostly vegetables?
Mostly vegetables, grains.
Expensive to
Does he
eat
cook-
meat in India.
Does he cook the veggies?
So most of what he eats is his wife cooks. Actually, he doesn't do the
Kind of
cooking.
place,
Speaking of
what
wife-
kind of place does he live in?
Oh, go ahead.
I'm sorry, Dickson.
No, that's good. So he lives in a concrete house.
Or
Do
any-
you guys remember that? That's like slightly better than a thatched roof.
Yes,
Yeah,
yes, we do
but,
remember.
So he lives in a concrete house. No screens or anything
No
on the
screens.
windows.
Is
No,
he the
no
only
screen.
one in his family suffering from all this?
There are other people in the area who have similar problems.
I mean, of his family members.
Of his family members, he says he's the only one in the family
Okay. But there are other people in the Assam area that have
that
this
have
similar.
problems.
Right.
Mhm. And where does he get his water from?
So in the little area, there's a pump, and so they go to this area with the plastic jugs and they fill it from the hand pump.
Does it have a handle on the pump?
As a
I'm just
handle.
kidding.
Yeah. No. So no, it's the.
Does it have a bottom on the jar?
So there's the hand pump.
Remember, Dickson, the story of dracunculitis, where they put their feet in the
I do.
drinking water thing?
Who could forget? Who
Yes.
could forget? They scoop up the water. That's right. But that's not the case here. And that's part of the reason they installed these pumps, to prevent them.
Mm-hmm.
But they traded it off for some other problems,
Of course.
which we could ask about, I guess. But
What are the problems? What do
you
you
may not tell
what do
us.
you- what do you-
Well, one of them is arsenic in the water,
oh.
which does cause a discoloration of the skin and a brittling of hair and the cracking of fingernails and stuff like this. How did he look otherwise? Any of those symptoms?
So we're
Signs?
gonna give you his exam in a moment
Okay,
if you
fine.
ask more questions. But it is interesting. So if you think about where the Himalayas or the Himalayas are, they border that northeast part of the triangle. So the issue with the arsenic is a lot of the rock. There's a leaching of the arsenic into the groundwater. So I think this came up in some article we read at one point. But I would not be surprised if there was arsenic in the water.
Yeah.
He
But
does-
remember, this week in parasitism.
He doesn't travel, right? He's been here.
No,
his
he's
whole
been,
life.
and that's been the issue. He's been in Assam sick, and he finally got the funds to come to this regional medical center for evaluation.
Hey, Dickson, what else you want to know?
Well, I mean, of course, after the physical exam, and he must have been a little bit underweight, I think,
So he's a little bit underweight. He's a thin man.
because he's suffering from fever every other day.
Does he have pets?
He does not. There are dogs, though, in the area he mentions, which he avoids.
How's his
I think I tell a story on TWiP where I was in the airport in Delhi, and they have these dogs, like the petting traveler's dogs, to relax and comfort you.
Yeah.
And everyone's got the wide girth. They're staying away from the dogs because the high degree of rabies in this area, and the dogs are just out and about. So there are dogs, but they're avoided.
No sheep or cows or
No, there's definitely cows.
monkeys,
There are monkeys.
Sure. That's right.
bats. Any bats?
He doesn't tell us about bats. I don't know about the bat.
Rats. I know there are rats too, right?
Yeah, there's.
And how's his appetite?
Fair. It's okay. It's not great.
On the days that he has fever, I presume that
When he has fever, not so good.
not so good. Right. And what is his fever?
So let's get to the exam, right?
Okay, yeah.
So he is febrile. His temperature is greater than 38 degrees. And on exam, it's interesting, and I have a picture of him here. You can particularly in the face see a darkening of the face, but you've got to ask him because people in a lot of the world vary in the skin coloring. So the issue for him is the change over time. I mean, you might look at this man and say, "Oh, he happens to have darker skin," but it's been increasing over time, and you notice in the extremities and the face, it's more so than on the trunk. So there's an increase in, and then as you go down, you see that when you look at what should be the whites of the eyes, they're yellow.
Right. So he's got some jaundiced.
So he is jaundiced, I would say.
So liver involvement?
So it's difficult to see on the skin. I'll give you a little bit of lab work,
Right.
but just a little.
Haven't finished the physical yet.
Yeah, we haven't finished that. And then what's really striking, and I have sort of jotted down here, I examine the man, and he has a very large liver and a very large spleen. Yes.
How did I guess that?
A very
How did I
large
guess
liver
that?
and spleen.
Hepatosplenomegaly.
Yes. And
No, you showed me that before, and I'm more sure of this case history than I was the last one. Although I could still be wrong, because you can't be definitive about this until you do the right tests. So, but this is
and we do some blood work, and I will let you know that his bilirubin is up. He does have an elevated bilirubin. And that's all I'm going to give you. We did a bunch of other testing.
I'm sure you did.
We will
we do ALT.
We
liver enzymes.
Liver enzymes. So there's the AST and the ALT.
I see.
And there is some increase of those.
Okay.
And I think is that enough? Any other questions?
Tons of information.
I think that's good.
Tons of information.
Okay. A lot of insect exposure. I think we mentioned a lack of screens.
No screens.
No screens.
Sure.
It's not protected against that.
Sure.
Does he sleep under a mesh?
No. He does not have mosquito netting in his area.
Okay, no mosquito netting.
But it's an every other day fever.
And it's been going on for 16 years. Because, there could be something else. But
16 years.
Yeah, it's a long time for every other day. That's a long
What does he do? He stays home all day?
It's agriculture.
Right.
He's a farmer.
They work out in the fields, farmer.
Yep.
Does he still work out in the fields?
He does, but he has a tough time. He doesn't have a lot of energy. And that was actually part of the problem.
Has he got any lesions on his body anyplace?
None that we see. And actually we do. We do the head to toe. We're not seeing any.
No scarring or where a lesion might have been?
I think that's the tough thing, right? Is that he definitely has, scars here and there because he's working out in the fields. It's a rural area.
Okay.
But I can't say that I see anything. I say, oh, look at that scar.
Right.
The fields are not wet, are they? He's not with his bare feet in water all day.
Oh, yes. That's the problem, right? It rains. Most of them can't afford shoes, so there's a lot of barefoot.
And what is the predominant?
But I will say he's sort of lucky. I mean, we saw a lot of fungal foot infections there, the Madura foot stuff.
Yeah.
His feet actually, they're calloused, but there's no...
Madura meaning elephantiasis-like.
Well, Madura actually is a town, right, in India.
Right.
And so the Madura foot is this chronic either higher-order bacteria or fungus, or there is a variant that's a staph variant. But it's I have some horrifying photos of Madura foot.
What was I going to say? So barefoot in the water. I had another question having to do with that.
Rice? Maybe rice.
Oh, what do they do with human feces in this village?
They do exactly what you think they do.
They just they just leave it out there, and it gets into the water and the fields and everything.
Yeah,
along with the animal feces.
Well, the animal feces, it's everywhere, and the human feces is equally poorly.
Right. What do you know the specific town he's from?
I don't actually. I don't have the town. I just know that he's from Assam.
No.
So I will just sum up what I feel are the key.
Okay, I believe you.
Okay.
I believe you.
Well, I will sum up.
I've just read that book thrice.
Yeah.
Yeah, I'm sure you did.
Okay.
Go ahead.
So I will sum up the key findings in this case. Okay. So as mentioned, this 16 years of fever. He has darkening of the skin and jaundice. He has enlarged liver and spleen. And he is from this inland area of Assam. And though he works in the fields as a farmer, we don't have a story of, just to, I think, help people, we don't have a story of him in freshwater streams or lakes or things like that. It's muddy areas where he's working as a farmer.
Right, exactly.
And a chronicity, a fairly 16-year chronicity. I think that's enough. I think people, when they think about it, they'll get it.
A lot of information here.
Plenty there, yes. I think the skin is a big thing.
And then we're going to do some tests, and we're going to diagnose and treat the man. He's going to...
Good-o. Yeah, and he's going to get better, too, I bet.
We'll see. Well, we have to find out what he has.
No, but I trust your judgment on this.
Okay.
Just give me the drug.
All right, we have some emails, Dickson.
We
Perhaps
do.
you and Daniel may share his laptop.
Do you want to go first?
Yeah,
actually?
you guys
do you want
can-
I would love to go.
This
Actually,
is-
no, I'll go
the
first.
first one is-
I want to go that
So the
first.
first one
Okay.
is the first one is a differential from last time, but it's still useful because Yosef puts work into it.
Now, did Yosef? That's how we pronounce it, Yosef? Did
He said at the end that we do a good job of pronouncing.
Oh, okay. So we'll stick with,
Yosef.
He said it's basically the same way as Joseph.
but just start with a yo. So
We have a Yossi.
Dear TWiPtasomas. My first guess for this case would be Schistosoma mansoni.
This is the missionary in Lake Victoria,
Yes,
right? Yes.
And I believe that was correct. 6 to 8 weeks after initial infection can lead to Katayama fever, which can present with dyspnea, rash, and eosinophilia. I would like to see the rash in order to make a more accurate prediction, and also know whether or not the rash was itchy. The parasite is obtained by contact with infected freshwater, and the cercaria burrow their way through your skin into your bloodstream. Treatment with praziquantel. And I think just to mention, we mentioned that the rash that we're seeing in Katayama fever is not a penetration rash. This is my I'm adding. And so the penetration rash would be in areas exposed to the water. The Katayama fever is more of a diffuse inflammatory rash. My other guesses are strongyloidiasis. Strongyloides do live in Lake Victoria and cause all of the symptoms of our patient. I would be more confident if the rash is serpiginous. I am uncertain of the time frame. However, if the infection was asymptomatic at first and got worse due to autoinfection, then maybe that would explain the late appearance of the symptoms. Treatment with albendazole. Necator americanus or Ancylostoma duodenale, similar reasoning to the strongyloides. It is also possible that he could have co-infection of 2 or more of these worms, and so may explain his extremely high eosinophilia. I would want to get stool samples to determine exactly what he may have. Non-parasitic causes, Scherg-Strauss. This is potentially very dangerous, and one of the things at the top of my list if he starts to develop granulomas. I think we've renamed that because maybe those guys were Nazi physicians, by the way. What's just to say.
What's the new name? Do you know?
I think we call it a, what do we call it? This is horrible. Eosinophilic granulomatosis might be the current name.
Okay.
I will look that up and jump back in when I know that for sure. I would just double check the CBC. Allergic aspergillosis, a history of asthma or cystic fibrosis, or another chronic lung condition. There also shouldn't be a rash with this diagnosis. An allergic skin test, just like TB, can be used to diagnose. Sincerely, Josef Davidoff. And then he says, P.S., you guys pronounce my name fairly well. You can pronounce it the same way as Joseph. Thank you for all your great work. Joseph.
Good. He's the guy at that Long Island Medical School.
Yep. Do you want to read next?
What is it? Hofstra Medical School? Is that what it's called?
It is Hofstra Northwell School of Medicine.
Got it.
Interesting. Anthony writes, if memory serves me correctly, with identical twins raised in the same household, the chance of one developing schizophrenia if the other one does is very high, but not one. That is to say, it's not a done deal. For identical twins raised in different households, the chances of one twin developing schizophrenia if the other one does is still high, but considerably lower than for 2 raised together. One might argue that schizophrenia has a strong genetic component that's necessary, but not sufficient. Again, if memory serves me correctly, there are a number of environmental stresses known to be associated with schizophrenia. For the speculative leap for someone who has the inherited predisposition for schizophrenia, perhaps a toxoplasmosis infection might serve as the stress trigger. Just a stray thought.
Well, let me jump in. So I did look that up, and yes, they've renamed Scherg-Strauss syndrome to eosinophilic granulomatosis with polyangiitis. Okay. And if I remember correctly, it was that I think these guys were Nazi physicians, and we've tried to like Reiter's syndrome and things that basically people who
Clara cells.
Yeah, try to sort of remove their names from the
purge.
Mengele's whatever.
Yeah.
Good.
So the schizophrenia and toxo, we talked about that last time.
We did.
It's people are really talking about this in the news.
John Boothroyd really was very adamant about it.
I know, but maybe
he's a world expert.
Maybe he needs to reconsider.
Maybe he does, but maybe I should call him and find out whether he has.
Why don't you do that, Dickson?
I will. I'll get in touch with him. No, I will.
Yeah, I'm serious.
Because, he's a very conservative guy. He's from Canada, for gosh sakes.
Really? They didn't be conservative?
Yeah.
So you would consider yourself not to be conservative? Is that correct?
Well, no, but it's not a belief system, remember?
Yeah,
Science
it's not belief. You're
system.
right.
So you keep an open mind, but it's not done on hearsay. Okay.
Well, so far, what I have seen is an association, not causation.
Right.
So we need more data, but I don't know how you get that data. Very difficult. Some things go on and
Somehow
on forever
you have
because
to
you
be
cannot
lucky.
get the data. Well, chance favors the
You know what would be
prepared
great if the twins acquired toxoplasmosis at the same time and then were separated, and one got schizophrenia and the other one didn't.
mind. You can't do that. That's not ethical.
But that's an accident of environment, and that might have happened already.
Maybe. Anne writes, Dear TWiP team, thanks as always for the wonderful podcast. I enjoyed the discussion of this week's paper on social organization in trematodes, in part because of the revelation of the still extant divide between macroscopic field work and our increasingly molecularly based laboratory work. The discussion arose regarding access to a microscope and camera. I think Daniel's observation that the lab strain of snail was the only one to differ in biology from the field isolated species is particularly interesting. Dickson's interjections regarding integration of general biologic ecologic perspectives in many of the discussions is an important reminder of the real-world pertinence of the topics presented. Now, explain to me why when you interrupt. Is that important?
I didn't write that.
There are so many things we can only study in the laboratory environment, but if we lose the connection with the real world, then the work can be elegant but may be less useful or even misleading. The comment is not regarding basic science, but the use of appropriate models and understanding of their limitations. I understand that. I think it's true that we need to continue to do these almost observational experiments in nature, right? Even though they're not always in favor.
I mean, climate change issues, for instance, those are all observational.
Yeah, that's right. Off my soapbox now. I spent several years studying wildlife biology before starting my professional education and still miss it sometimes, although I don't miss it when it is cold, wet, buggy, or humid, i.e. not often. This is Ann again, a DVM PhD from Beaverton, Oregon.
Right.
Now we have Victor. Victor writes, this is a message from the long gone past concerning TWiP number 37.
My goodness.
Dracunculiasis. Dickson comments on how Sweden was planning on building a 17-story vertical farm, and you, Vincent, ask if Dickson said Norway. Dickson then says you shouldn't mix those two since we don't like each other. As a Swede, this is far from the truth. I imagine it's similar to the USA's relationship to Canada. Oh, you mean our 51st state. Lots of tongue-in-cheek jokes between the two, but it's more of a sibling thing. At the end of the day, you're really happy you're neighbors. Besides, Norway has better skiing slopes. Anyway, I found out about your This Weekend suite about 3 weeks ago, and my brain is starting to hurt. I'm a freelancing artist, which means I spend hours upon hours at my desk drawing and resting my forehead on the desk trying to summon inspiration. This also means I listen to hours upon hours of podcasts. Your podcast is the perfect mix of learning and humor without letting the jokes take the front seat, which sadly is the case with many podcasts. I don't want to ask questions since I have a feeling that my 550 episodes TWi backlog will answer them. But as someone who likes to draw things from nature, do you have any recommendations for parasites or viruses and bacteria that would look good on an illustration? Probably not going to be a big seller, but I like to do some personal work for my own enjoyment. Thanks for taking time out of your lives to share your wealth of knowledge. Victor.
What should he draw, Dickson?
Well, it depends on what you can see. I mean, some of these things are pretty darn small. So if you haven't got access to a microscope,
he could look for pictures of them.
Then, no, you can look for pictures. It's okay like that. But I mean, a reduvid bug, you can see them and they're big and they would make a nice drawing. I would love to see a reduvid bug.
Maybe life cycles, right? I mean, I think it would be nice
Or
to
life
have
cycles.
some.
Yeah.
And because life cycles are nice, you get to draw the vectors, you get to draw the microbe, you get to draw the environmental context.
Daniel, you're absolutely right. But every one of those drawings in our textbook
Yeah.
was drawn from real life. So I had to actually bring the specimens to the artist, and he had to see them under the microscope before he would agree to draw them. He says, I'm not going to draw anything that somebody else observed. I want to see it for myself. So that's really firsthand experience.
If it were up to me, of course, I would draw a virus.
Yeah, but
I think
That's
the schistosomes look pretty cool.
Schistosomes are cool.
I think the adults are cool, and the cercaria are pretty neat.
They're all neat stuff.
So that'd be my suggestion.
If you had
have
a
any
microscope, you could go to
suggestion?
town on a lot of this stuff.
Yeah, I think the flukes. I think a lot of the
flukes are beautiful.
The flatworms are pretty cool. Yeah.
Especially the big ones. I mean, you can see them with your naked eye, and they have beautiful patterns. Lots of symmetry in nature, and a lot of these are like that.
Do you want to take the last one, please?
Sure.
John writes.
Vince and Dick. Sorry about that, Daniel. Love your show. Do you know what effect fasting would have on strongyloides? That is, could strongyloides be starved to death? For example, a 40-day fast? My goodness. The answer is, there's more to this.
No, just thank you for your work.
That's it.
And it says, thank you for your work. And I have an answer. If you can go without food for 40 days, that's a very long time. But remember, strongyloides doesn't eat your food. Strongyloides eats your tissues. So it would live as an adult embedded inside the columnar cells of your small intestine. I don't think their nutrition would be upset at all by this.
I think it might be worse, right? Because
it might be worse. It might be worse.
You're starving, and your immune system.
It might be worse.
Yeah,
that's right.
It could be a problem.
And the larvae that migrate, they eat bacteria from the gut tract, and then they penetrate in auto-infectious cycles, at least. So, I don't think that nutritional limitations would have any effect on strongyloides. But if you were talking about Ascaris, on the other hand, which only eats pre-digested food that you ingest and then digest, those worms might starve to death and decide to move, find a new house.
How many days would it take?
Well, I think maybe less than a week.
So how long can you not eat a day? If you're assuming you're drinking a lot of water, how long can you live and not eat?
A lot of it has to do with your fat stores, right? So
I could live for a year and a half like that.
Okay.
I'm willing to make fun of myself. I really am, because I know I'm a little bit overweight. Okay.
No, Dickson, you might only last 9 months.
Thank you, Daniel.
No, actually, some of the worst cases of strongyloides I've seen is malnourished children. And so I think that the lack of eating, the malnourishment, can be quite a problem.
That's right. And some of these worms cause that. I mean, in fact, Ascaris secretes an antitrypsin factor, which prevents you from accessing your own food stuff. So either way, you lose, I think, on this one.
Okay, thank you, Dickson. Thank you, Daniel. This is TWiP, which you can find on iTunes, microbe.tv/TWiP, or you can use any of your favorite podcatchers to subscribe. You can find the link at microbe.tv/TWiP. Please send us your questions and comments to twip@microbe.tv. Daniel Griffin is here at Columbia University Medical Center. Thank you, Daniel.
Oh, it's been a pleasure, as always.
Dickson Despommier is at Racaniello.org. Thank you, Dickson.
You're welcome, Vincent.
I'm Vincent Racaniello. You can find me at virology.ws. The music you hear on TWiP is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP
is parasitic.